Hello, everyone. I'm going to be talking today a little bit about mastering the TRA through better chemistry, specifically discussing strategies to increase the analytical evaluation threshold. Start off, my name's Kevin Trankler. I work here at EAG. My capacity is as the Director of Analytical Chemistry. I was brought on board really to kind of expand into the medical device testing industry about 3 years ago. My primary responsibility is over medical device testing with a heavy emphasis on 10993 Part 18, but also we do a lot of investigational analytical work such as contaminant ID, particle analysis, and some method development validation. We've recently started to expand into in vitro testing at a different site. My background is I'm a chemist. I got my PhD over 20 years ago. I've been in the industry for more than 20 years, primarily focused in analytical chemistry. I have a strong quality background. I led a quality team, global quality team for about 7 years. Strong understanding of the ISO standards, GMP, GLP, but primarily I've been focused the last 3 years on chemical characterization. So I do want to emphasize that this talk really reflects the current interpretation of the standard. This has evolved significantly over the past 3 years. I think there's been a lot of changes in expectations, and we've seen a lot of that through our interactions with FDA, but also through client interactions and their interpretations of FDA requirements. I suspect these will continue to evolve, and there could be additional changes in the future. So I just want to emphasize that this is the current status as of now and kind of how we interpret the standard. So just to kind of start out, I want to give an outline what I'm going to talk about today. I'm really going to start off with a description of what the analytical evaluation threshold is and why it's important. I think there's a lot of people that have had a lot of exposure to the AET, but maybe some that haven't. So I want to make sure that we're working from the same basic understanding from the start. Then I'll talk somewhat about, minimizing extraction volumes, qualifying the materials that you use to extract the devices, custom extraction setups. I'm going to go into a little bit of a discussion around the dose-based threshold and the importance of understanding what your dose-based threshold should be and justifying what that dose-based threshold is, what that impact is on the AET. A little bit about reducing uncertainty factors with supporting data. Then finally, I'm going to talk what's remaining, and that's really if all else fails, you may need to concentrate the sample, implications associated with concentration, but also just generally improving sensitivity with your instruments. So to start off, what is the analytical evaluation threshold? Well, it's simple. It's a threshold that has been established through calculation that is the threshold you need to be able to demonstrate your sensitivity. So anything above the analytical evaluation threshold needs to be identified and semi-quantitated, and anything below it doesn't necessarily need to be identified or quantified. So in essence, your LOQ must be below your AET, and if your sensitivity isn't sufficiently low enough, then you may not be able to demonstrate that you can detect down to the AET. So that really is the sensitivity limit that you need to demonstrate. It's very important. It's calculated, the calculation is in the standard, and there's a few variables here. The AET is really based off of the dose-based threshold, and then some parameters around the number of devices, concentration, volume of solvent that you're extracting with, and then ultimately it's divided by the uncertainty factor. Some of these variables are variables that we can control to some extent. Others, we may not be able to control them, but we need to make sure we establish them correctly. Specifically the dose-based threshold, and C, which is the patient exposure. These two items we may not have a lot of control over, but if they're defined incorrectly, can lead to a lot of challenges around achieving the AET and may be overly conservative. So it's really important that we understand those, and I'll talk a little bit about that later in the presentation. I think as far as the rest, they all have some control. For example, A is the number of devices, and in some cases, we may have devices that are very small, so we may need to have multiple devices in there, which will increase the AT. But ultimately, if we can have more devices but hold B, which is the extraction volume, constant, we can achieve a higher AT. So another way to look at this is if we have one device and we decide to use less solvent than is suggested or recommended in part 12, we can actually increase the AT by using less solvent. It's important that if we do that, we really think about what the impact of that might be. For example, you need to have enough solvent to cover the device and fully submerge it, which is a big one. You also need to have enough solvent such that you don't saturate the extraction solvent. So if you have too little solvent and you saturate it, you will not get effective extraction of the extractables. Now, that's pretty easy to circumvent if you're doing an exhaustive study, because in an exhaustive study, ultimately, you're going to demonstrate exhaustiveness. And so you're not concerned about saturating the solution because you're going to continue to extract until you can demonstrate that less than 10% of the initial extract is coming out. So you're going to hit the same endpoint. It may take more cycles if you potentially saturated the very first extraction cycle. But generally speaking, you can control B. There are some things you need to consider. For example, you need to make sure you have enough solvent in the end to actually analyze through all the techniques. So if you have 4 or 5 techniques that you're going to do analytical work on, you need to have enough extraction solvent to be able to subdivide it among those techniques. The other one that you can control to some extent is the dilution factor. Obviously, if you're using less solvent than is suggested or recommended by part 12, you wouldn't be diluting it, but there might be cases where you actually concentrate that extraction solvent further after you've done the extraction, and that would result in a higher AAT. The other thing that you can control to some extent is the uncertainty factor. There's a lot of literature out there that really has come to some level of agreement on what is a industry accepted uncertainty factor for LCMS and GCMS, for example. They're rather conservative, and if you put the extra time and homework into actually evaluating your uncertainty factors, many times you can get these numbers lower by doing it. You have to build a good database. It needs a lot of input on it, but ultimately that uncertainty factor could reduce the AT. So extraction volume. I talked a little bit about reducing the extraction volume on the front end so that you can effectively increase the AT. And there are recommendations in part 12 related to the surface area of the device and what is the recommended surface area or solvent to surface area ratio in order to achieve good extraction efficiency. You can diverge from that though, especially if you're not concerned about saturation, which I mentioned earlier. And when you do, you will effectively increase your AT. Now, there are a lot of things you need to consider when doing this. I kind of touched on some of those, but the big one is really making sure that that device is fully immersed in the extraction solvent. And I think sometimes that can be challenging, especially if you have odd-shaped devices or very linear devices, and you need to come up with creative ways to ensure that those devices remain in solvent contact for the entire extraction time. The picture on the left there is a suture or a thread that was floating on top of the extraction solvent. So you may also have to find ways to actually fully immerse it, hold it underneath the extraction solvent, which is the case here. But when you do all of these things, you need to make sure that, one, you're getting good solvent access to the device. You may need to consider secondary containment, ensure that the device is fully sealed and you're not getting any leakage of volatile solvents. And some of these, creative extraction vessels can sometimes be very challenging to achieve that. Talking about that a little bit further, the case on the left here is a very narrow device, and trying to submerge that in a jar with minimal solvent can be very challenging. So, we found that using PTFE tubing can be one way to get around this challenge. You're really maximizing solvent contact. Filling up that tube is a lot easier than filling up a jar, and you have a lot less solvent than if you try to fill up a jar and submerge this device. The other thing that many people have done, including us, is use solvent displacement as a means to increase the solvent contact with the device. But you have to remember that there are some challenges associated with this. So typical strategy would be using glass beads to displace the solvent. It's really important that you use borosilicate glass so that you're contributing minimally to that extraction setup. But there are some other problems that can happen. For example, the glass beads can abrade the device, resulting in device degradation or contribution you wouldn't see under normal extraction conditions. The glass beads themselves can oftentimes contribute to the residue. They have coating, they can flake off glass. You will get a large silicon contribution in your ICP-MS data, for example, boron as well. Generally, you can pre-treat these devices, these added displacement tools that you're using, and try to minimize their contribution to the extractable profile. Another good strategy is Teflon bags. These Teflon bags are available in different thicknesses, and in some cases you can heat seal them. I wouldn't suggest doing that with flammable solvents, but and in those cases where you can't heat seal, you're going to need to find good methods for sealing that device. You also, or that enclosure. You also want to make sure you're not leaving a lot of opportunity for headspace to swell up the bag and allow that solvent to go into the volatile phase. But there are a lot of creative ways, and I think the key here is remembering that the extraction setup is really something you need to look at in order to help improve the overall contact area with the solvent. Now, it's important when you use some of these creative extraction vessels that you understand their potential contributions to the extractable profile. Oftentimes they're not necessarily available from cleanest manufacturers. There may be different manufacturing processes that leave residues, or cleaning processes that are done on them that may leave residues. So it's really critical that you pre-evaluate these devices and really understand what the potential contributions are going to be to your end extractable profile. And in those cases where there are significant contributions, you need to really be concerned about it, because in some cases those contributions, in the case of LCMS, for example, could lead to ion suppression and an inability to adequately detect or quantify analytes, extractables that might be in that same region. You could subtract out the contributions from the device, from the extraction vessels, but ultimately they could have a significant impact on the quality of your work. Now, to get around that, you can continue to search for cleaner sources for those devices, for those extraction vessels. Alternatively, you can pre-extract them, precondition them. Develop a process around cleaning them and preconditioning them in the solvent that you're going to be extracting with, and then you'll circumvent the problem of contributions from the extraction vessel. The other thing to think about with custom extraction setups is really can be challenging, especially with complex devices, where you might have a certain region of that device that is patient contacting, but other parts of the device are not, and you really want to limit cutting of any type when it comes to device analysis, because it can lead to other contributions in the extractable profile you wouldn't normally see. So you may have to come up with creative ways to just in case the part of the device that is in contact with the patient for the extraction. Now, in some cases, you can argue, well, the whole device is manufactured under the same conditions. And in those cases, if you can defend this and you can document that your manufacturing process is consistent across the entire device and that the sterilization process is the same, then you can really justify extracting the whole device and treating it as more than one device. So say a 12-inch long device, but only 6 inches is in patient contact, if you can justify that that whole 12 inches is manufactured in the same fashion, exposed to the same processing aids or cleaning agents or sterilization technique, and document that, you can treat it as two devices. And then you effectively, if you use the same amount of solvent, you double your AT. So being creative around those things, those strategies, and trying to defend that and justify it is a really effective way of improving your chances of success. Now, I want to talk a little bit about dose-based threshold, because I think it's important that you do not take the determination of what your DBT should be very lightly. And I would encourage you to make these decisions under the guidance of a toxicologist, because this is a pretty impactful value. It extends across 100 times, so 2 orders of magnitude. So if you were to incorrectly determine that this should be a 1.5 because it's long-term and greater than 30 days, and really it could have been justified that it's a 20, that's an order of magnitude difference. It's 10 times lower AT that you would be stuck with than if you had made the correct assessment at 20. There are some cases where it's not always obvious or clear what the patient exposure time is. I can think of a couple examples, but one example would be in the case of a device that you're evaluating that is really the device that's used to manufacture or, say, grow what is ultimately going to be the true medical device that the patient is exposed to. And that ultimate end-use device is actually a permanent implant, but it's really being manufactured or grown in this secondary device, such as, maybe an externally communicating device, and it's only exposed to that device for a brief period of time. Yes, the end result is put into a patient, but the potential extractables that it comes in contact with that could then be transferred to the patient is actually a much shorter period of time. So your dose-based threshold for that evaluation should be higher. But you may get caught in the trap of thinking that the end-use device is really what we're talking about, and really that wouldn't be the case. So I think it's really important to talk to a toxicologist, make sure you understand your device very thoroughly, and really describe how the end use is administered to the patient. I think the other thing that oftentimes people don't think about are resorbable devices, or maybe don't think about them adequately. And this is really where you do want to consult a toxicologist. But if you're able to defend that, these resorbable devices degrade in a certain period of time, and that you have some supporting implantation study that you can also defend the safety and biocompatibility, then you really shouldn't be thinking about it as being exposed to a patient for a lifetime, because really they're exposed in a much shorter period of time and they're fully absorbed by the patient. And so in those cases, you can potentially defend using a higher dose-based threshold and ultimately having a much higher AT and a big impact to your study, because these resorbable devices can contribute a lot to the extractable profile. So the lower that you have to go, the more difficult it's going to be to pull some of that data out from your extractions. So again, consult with a toxicologist, understand your device, and come to a reasonable dose-based threshold. Now, let's talk a little bit about uncertainty factors. I think there's a lot of information out there, and there's been a lot of discussion around uncertainty factors. And I believe that there's a good position to be in by generating your own data to support uncertainty factors for your instrumentation and your method. We've read literature that suggests LCMS can have a uncertainty factor of 10, and it's kind of commonly accepted and really isn't challenged if you use 10 in your calculation. Most people believe it to be conservative enough. Ultimately, though, if you collect data under controlled experimental conditions using a validated method that you validated, and you collect that data in the same fashion, you should be able to demonstrate that your uncertainty of your instrumentation uncertainty factor is much lower. Certainly half an order of magnitude lower for LCMS in most cases, and maybe not quite as impactful for GCMS. But that half an order of magnitude buys a lot of real estate when it comes to increasing the AAT. It's important that when you develop your uncertainty factor database, your relative response factor database, the selection of the analytes that you put in there makes sense and are representative of a wide diversity of potential extractables. And decisions about that should be made based off of a lot of factors. You might have chemist input, people that have actually interpreted data, seen what comes out of some of these extractable profiles, will be able to make logical decisions about what should be included in the relative response factor database. You should be evaluating materials of construction. In our role at EAG, we don't really get the opportunity to say we're only going to work on one type of device. We see a wide diversity of devices, but in some cases, in certain industries, you're doing your own internal study, you may only really ever see the same types of materials of construction across the devices that you use. And in that case, really having a good, relative response factor database for those types of potential extractables is invaluable. You can also resort to looking at the literature and seeing what might come out based off of other people's experience. That's the one luxury that we do have here at EAG, is that we do get to see a wide diversity of devices and see a lot of different extractables. So we've really been able, fortunately, to develop a pretty robust database. Ultimately, you then need to collect the data. And I think one of the things that may sometimes not happen is that people may not necessarily collect that data under the same conditions that they're running extractable studies. So, you want to have a nice multi-point curve. You want to be able to evaluate the linearity of that curve. You really want to know that you're collecting the data under the best case scenario using a validated method so that you can depend on that data long term. Now, ultimately, if you can't really affect the AT low and high enough and are not able to hit the sensitivity required for your AT, you're left with only one option, and that is to concentrate the sample. And when you concentrate the sample, you can effectively increase the AT by, reducing that dilution factor. So it's the inverse of the dilution factor, and that has a proportional effect upward for the AT. It's important when you think about doing this that you really plan this out on the front end of the study, because if you wait until the end and you decide you need to concentrate the sample, you may be stuck in a situation where the level of concentration that you need is so great that you don't have enough sample to analyze in the end. So you need to kind of target what is your minimal amount of solvent you're going to need, extractables solvent that you're going to need for all the analytical techniques, and then really target that as your endpoint. So if you need a 10-fold concentration, you need 1 mil, you need to make sure you're starting out with at least 10 milliliters of solvent, or you won't have sufficient solvent to analyze the samples. So it may require more solvent, may require more devices in order to hit that. The other thing that you will need is you'll need to be able to defend that concentration step. So one, it needs to be very mild conditions. You need to have it well controlled. And I would encourage you to validate that concentration step. So, you can repeat it and use that same approach in the future. This is all important because ultimately you'll be required to demonstrate through spike recovery experiments that you get good recovery on, a diverse set of surrogate standards. Because ultimately the big concern is that through that concentration step, you may lose extractables, or you may alter those extractables in a way that they're no longer representative and cannot therefore really be assessed by a toxicologist. So you need mild conditions, it needs to be well controlled, and you need to demonstrate that that has minimal impact on the extractables. Ultimately, the last thing you can do to increase the AT, well, you may not be able to increase the AT. The only thing left is decreasing your LOQ. And so the important thing here is to really make sure that all of your instruments are performing well. You have a good system suitability experiments to ensure that they're optimal, operating under optimal conditions, and then validating those methods so that you really have good control over them. You know, untargeted screens are really completely, they're very different than targeted screens. You have a lot less control. It's relatively straightforward to set up a targeted quantitative method, but having a good validated method for a wide, diverse set of compounds that you don't even know what you're going to see is very challenging. And so you need to keep that in mind when you're looking at your methodology and validating it, and really ensure that you have a good set of system suitability parameters that you know your system's operating effectively. So we kind of talked over all the key parts about the AT, number of devices, concentration, solvent, uncertainty factors, the patient exposure, and the dose-based threshold. And although you don't have a lot of control over the dose-based threshold and the patient exposure, it's important that you come to a good consensus on what those values should be and that they're not overly conservative or unnecessarily conservative. The other parameters in green, you do have some flexibility with, not a lot, but you do have some flexibility. The important thing is to plan all of this on the front end, make sure you capture it all in your protocol, and that in the end, you know, you're really thinking about the end point. And that should lead to a good study. So thank you for your time.